Hey everybody, welcome back to HPC Tech Shorts, the engineering water cooler here in AWS. Today we're going to be talking about one of the many use cases that we see in the wild for AWS Batch, which is our high-performance computing cloud-native scheduler that schedules containers and tasks on AWS. But today, with the interesting use case, I'm joined by Brian Loyal in St. Louis. Hey, Brian.
Hey.
And Ujjwal Ratan, who's in Seattle. Hey, Ujjwal.
Hey, how are you?
So now you guys are solution architects in the global life sciences team, and you look after all sorts of, well, whole range of different workloads. But it's the global life sciences AI/ML team is really your specialty area. And we started having a conversation a few weeks ago about the work that you guys have been doing with AlphaFold. Which got everybody interested, and of course, I think everybody who's got cells is interested in what AlphaFold can do for most of the life sciences research that's going on in the world.
So this is essentially our goal as architects. We're responsible for really, defining the cutting edge for our customers, and this was kind of what we aspired to. We said that, hey, there's a lot of data scientists out there, or bioinformaticians, or just bench scientists. I used to be a bench scientist, who have a lot of really interesting questions about proteins. And for those in the audience that don't know much about this, so the proteins are essentially the molecules in your body that make your body go. So they're responsible for everything, for moving your muscles up and down, for digesting your food, for giving you energy. It's a really key part of what makes life. It's a really important structure. And the interesting thing about proteins is that their shape is really essential to how they function. The kind of metaphor we used to say was that, like, if you think about like a key and a lock, except that the lock and the key were both made of like modeling clay. It's this really interesting interaction, how proteins interact with other sorts of molecules in your body, to get work done. And so understanding the shape of these proteins and their structure and how they interact with other things can give you a lot of insight about disease. And so, there's a lot of customers at like pharmaceutical companies who are trying to design new proteins or new molecules to do things like, the next generation of antibodies for, to prevent the next COVID, or cancer therapeutics, or even just understand how, human health. It's a really important aspect of research. So what we tried to build was, say, if you are a scientist and you have a protein sequence that could have come from a variety of places, including some, bioinformatics techniques done on HPC and AWS, we want to have a system where you can work in a really familiar environment, like a Jupyter Notebook, and then you submit that sequence to some sort of service behind the scenes, where magic happens, at least from the perspective of a user, and then it returns this structure. And you can then use that structure downstream for all sorts of good workflows in terms of analysis and design. So we try to make this as easy as possible. Our goal here is to really, widen the base of the pyramid in terms of people who can experiment with this system. And we're pretty excited how it turned out.
So maybe it's a good idea to sort of talk about why, right? Like, why do we need to build this, or why should people care about this? Yeah. Aside from the awesome work that scientists are doing in the space of protein folding, right? What we saw as solution architects and machine learning, specialists at AWS is a huge gap between how the science has been progressing and how it's being utilized in real world, right? And where we see the gap is, even though the scientists are kind of pushing the boundaries and creating, repositories and knowledge about all of these awesome things, in this case, protein folding, there is still limited knowledge on how to take all of that information and bind it into some sort of a workable, application. And what we realize is the gap is mostly around engineering, right? Like, how do you package this? How do you run this? So that's where we began. And if you can quickly flash the architecture diagram, and this is that bit that Brian was talking about in terms of magic, right? Like, this is where the real magic happens. in a sense that data scientists who work on a familiar environment like Jupyter Notebook submit, a sequence. And what we've done is we've sort of broken down that entire pipeline into modular bits. Some of these are CPU driven, some of them are GPU driven. And Batch actually does a great job in allowing us to orchestrate this, right? Like Batch is a great service that allows you to choose your own compute environment. So we could sort of say that instead of chucking it all into a GPU, where all the GPUs may not be actually utilized, or chucking it all into a CPU, we wanted to kind of give a choice. And Batch makes it really, really simple to choose between a CPU and a GPU. So that's what we have done with those two queues. And it comes with all of the CI/CD built into it, right? So it has the code commit repository, the pipeline that allows you to push code into production. It's all packaged nicely with a CloudFormation script that you can reproduce and run in your own accounts.
And so you've actually got all of this sitting in a GitHub repo, right?
Exactly.
If I get that right. And in fact, I'll flash up the URL here, but I will put it into the show notes so that everybody can come and grab it themselves later.
Yeah, so that's pretty much what we've built. And I think it'll be a good idea for us to show you a demo.
Yeah, let's see this thing in action.
All right, sure. We'll see this thing go. And again, the idea here is that this is an environment, Jupyter Notebook, pretty standard data science tool that should be pretty accessible to most people who are working in this space. So the way this works is we have a cell here with a protein sequence. So in this case, this is a pretty small one. It has a code T1084. And we can run it. It'll validate the sequence and upload it into S3, which is our pretty standard AWS object store. And then we can submit that to AWS Batch. And the nice thing about using Batch is it gives you a lot of control over the actual resources assigned to your job. So if you are really cost conscious, you can kind of turn that dial down and use a small number of compute. If you're running like a very large number of proteins in parallel, you can dial that back up and you can get that kind of parallelization that you need. So here what we're going to do is we're going to use kind of a default set of CPU and memory numbers that we kind of put together. And this job is going to proceed in two stages. So the first stage is going to be a data preparation step where we're going to take our sequence of interest and then compare it to thousands of other protein sequences to kind of get a feeling for like the evolutionary history of this protein. That's one of the big features that go into this ML prediction is by comparing the sequence to other proteins. That is very CPU dependent. That does not use GPUs. And so we can run it on a CPU instance and save a little bit of cost, which is nice. And then the output of that goes into the second stage, which is the actual deep learning model. It uses a GPU to make that structure prediction. So the nice thing about this is that you're not paying GPU prices for a CPU task. We're really trying to be efficient with the way we use the cloud. So we can submit that, and that'll toss the information to the AWS Batch service. It'll spin up the resources you need, and it'll do that kind of task. This is going to take a little while, so I'm not going to sit here and wait for Batch to do its thing. So instead, what I'll do is I will download a sequence that I did earlier today from S3 and just show what the results look like. And again, our idea was to really put all this information into one environment so people to look at. You get a couple of information. So one bit of information, which is interesting to scientists, is to say, if you look at your sequence, what parts of it are what we call conserved across other protein sequences? And this gives you an idea about, do you have enough information in your large sequence databases to make an accurate folding prediction? So in this case, you can see this area in the middle of the protein has a lot of commonality between a lot of other proteins. So, just intuitively, you can think that we know a lot about this part of the sequence. And so the structure of the protein in this area is going to be pretty accurate because we have a lot of background information. On the other hand, on the edges here, this is somewhat unique. There's not a lot of other proteins we can compare to get that sequence information. And this is pretty standard for a lot of proteins where the edges will be a little bit more unique than the middle part. The other thing we can see is the actual structure. And so this is sort of the result of all this is the folded protein. And so you can download that it's called a PDB file and display it directly in the notebook. And here's what you get. So in this case, this is the protein of interest, and it's colored in terms of the confidence. So one of the nice things about AlphaFold that the DeepMind team put in there, the team that developed the algorithm, is they do give you the idea to understand which parts of the structure they have more confidence in than others. And so it's kind of like we saw in the MSA diagram above, the middle parts of the protein, so kind of in the middle of all these squiggles, are a nice blue color, which means that it's very, very confident. But the edges are less confident. The red in this case is a negative element. So you kind of get a feel for both not what the structure is, but how confident you should feel about it. The nice thing about doing this work in batch is that in addition to kind of managing the resources independently, is that you can do this at scale. So I just submitted one sequence for analysis, but if you are so inclined, you can loop through this and submit hundreds or thousands of jobs, and they will just queue up automatically until the resources are available. So we hope this is going to be a really useful tool.
You could dial batch in to actually go and harvest stuff out of the spot market.
Yeah, totally. It's great.
And you could get really quite aggressive and quite big scale.
Yeah.
This is really cool. So what can you use this for now? Now that we've got an understanding of what the protein looks like, what's the scientist in the lab going to do with this?
Yeah. So, when I talk to customers who are in this area of kind of protein dynamics or understanding, a couple of really interesting patterns come up. So one is just understanding what these proteins bind with. It's called protein ligand binding studies or interactions. So let's say that you had, you're a pharma company and you're coming up with a new molecule that is supposed to treat cancer. So it's an oncology med. The way that those drugs work is that they will oftentimes interact with proteins. And so being able to take a structure and put it through a simulation where you try to figure out how does this molecule interact with it can really help you understand, is this molecule a good candidate for a drug, or should I just abandon it and go on to something else? So that's one good example. The other thing that people have done with it is the process historically of getting these structures involves some pretty advanced laboratory technology, things either called X-ray crystallography or more recently cryo-EM. And that's a really expensive process. It's super complex. And there have been some work done to show that you can use this kind of simulated structure to either augment or even sometimes replace those kind of laboratory experiments.
And this is very similar to like in computational fluid dynamics, which is another area that we work in all the time. This is actually really similar because in that space, in aerospace, building a model, putting it into a wind tunnel, simulating what that aircraft wing or whatever is going to look like, or actually observing what that looks like in a wind tunnel. It's a hugely expensive operation, hugely time consuming. You can only really afford to iterate like, one or two models a year, probably at that scale. You can do 100,000 of them in a day in simulation. And so, of course, the actual silicon version of the problem is actually moving the science along a whole lot faster than the physical one is.
Yeah, and one of the things I would say is this area is so new right now. I mean, we're still sort of at the cusp of what we can do with this technology, right?
Right.
So previously, when therapies and drugs used to be developed, it was necessarily a trial and error process, right? Like people used to scan through billions of compounds and molecules and find out that one molecule of interest that they really want to take to trial and build a drug around it.
Right. And actually estimating, working out what the protein structure was, doing that protein folding simulation prior to AlphaFold. That it took really large supercomputers. It would take weeks to simulate a nanosecond or two of folding time, isn't it?
Yeah, it's a search operation. Like you're going and trying to find out based on your best guesses of parameters on what exactly would work. Where AlphaFold is actually giving you a structure which you can like base your analysis on. The other thing I would say is we are at that stage where we're trying to map this, right? Like we mapped the Human Genome Project, where genomics was so huge and batch is like a great driver of those workloads. Trying to like decode the human genome. I think we are at a stage where the shift is now happening to proteomics, where we are trying to map the protein sequences and try to identify all known proteins, which is a big ocean of unknown information that we are now sort of getting into.
Wow.
Yeah.
Well, this is the stuff that gets me out of bed of a morning. This is why I like doing what we do. We get to see scientists and engineers doing some of the most incredible things.
Take it for a spin, right? We got the GitHub out there, and we've got all the instructions needed to deploy it. Even if you're not sort of super interested in working with protein folding, I mean, this is a good baseline example for pretty much any sort of batch computing workload that depend on CPUs and GPUs, and you have to make a choice. And of course,
that Jupyter notebook that Brian was showing has an incredibly well-structured approach to how to submit batch jobs from a notebook like that. So it'd be pretty easy to steal that code for something else too.
Yeah. And
gentlemen, thank you for coming today. I really appreciate it.
Absolutely. Thanks for having us.
Now, if everybody else out there, if you've got some ideas of things that you want to see us cover in future Tech Shorts, come find us on Twitter. Our DMs are open. We'd love to hear from you. Until then, see you all next week.